Greetings everyone. So now we're going to discuss about our next topic, which is the structure of nephron. So when it comes to the structure of nephron, we need to understand what the nephron is. And actual nephron is the basic structure and functional unit of kidney, and that is basically present in 1 to 1.3 million nephrons are present in each kidney. It is not a cell; it is a structure. It's made up of multiple cells. So basically, nephron is 1 to 1.3 million in each kidney. And when it comes to the type of nephrons, there are two major types of nephrons. Number one type of nephron, which is present in the cortex region only, is known as the cortical nephron. That cortical nephron is present 15% in kidney. That is 15% in kidney. And on the other hand side, it has shorter loop of Henle. Shorter loop of Henle is present in the shorter loop of Henle is present in the cortical nephron, due to which the loop of Henle does not extend in the due to which the loop of Henle does not extend in the medullary region, as well as the counter current mechanism, which is an important mechanism to produce concentrated urine, does not occur within the cortical nephron, due to which the concentrated urine is not produced in cortical nephron, right? And on the other side, if we talk about the juxtamedullary nephron, which is the other type of nephron which is present in cortical region as well as in the medullary region. Juxtamedullary nephron is present in cortical region as well as medullary region. It is found in 80. It is found 85%. It is found 85% in kidneys, and it has got a longer loop of Henle, the which extends into the medullary region, such that if we talk about this structure, so this is what we call as the renal corpuscle, after which we have got PCT for proximal convoluted tubule, after which we have got the loop of Henle, after which we have got the distal convoluted tubule, and finally the collecting duct. We're going to understand their function as well as their structure line by line. So when it comes to the collecting duct as well as the loop of Henle, both of these lie in the medullary region in the juxtamedullary nephron, while rest of the structures such as PCT, DCT, and renal corpuscle, all of these three structures are present in the cortical region. So juxtamedullary nephron has PCT, DCT, and renal corpuscle in the cortical region, as well as the loop of Henle and the collecting duct in the medullary region. On the other side, if you talk about its function, it has got longer loop of Henle, due to which counter current mechanism is possible. Counter current mechanism is possible, and which is a mechanism for producing concentrated urine, due to which concentrated urine can be produced in this, right? Concentrated, yes. On the other side, if we talk about if we on the other side if we just talk about the process, that what the nephron is divided into. A nephron is basically divided into two major structures. It is nephron has two parts. One part is the renal corpuscle. Number one part is the renal corpuscle. Number two part is the renal tubules and renal corpuscles. When it comes to the renal corpuscle is basically the renal corpuscle is basically made up of two components. Renal corpuscle, or we also call this as the Malpighian body, that is having two major components. Number one, as we have previously studied, that the renal artery drains into from the abdominal aorta. There is the dividing branching of renal artery, and each renal artery enters into each kidney. And once a renal artery enters into each kidney, that renal artery further divides itself into afferent arterioles, which further go into the nephron. And when they further go into the nephron, the when the afferent arterioles further go inside the nephron, then they further divide into smaller diameter capillaries tuft, or a smaller diameter tuft of capillaries, which contains around 40 to 50 capillaries, which is known as the glomerulus. Which is known as the glomerulus. And that glomerulus is a tuft of capillaries, which is a tuft of capillaries which is formed by the division of afferent arterioles. And that tuft of capillaries, glomerulus, is present in a specific capsule, which is known as the glomerular capsule or the Bowman's capsule or the glomerulus capsule, named after Bowman, who discovered it. And on the other hand side, if we talk about The structure of Bowman's capsule. The Bowman's capsule is basically lined with special types of cells. The glomerulus are actually lined with special types of cells. The glomerulus is lined with special types of cells. The glomerulus is lined with special types of cells which we call as the podocytes. What is the function of podocyte? Podocytes are basically the cells which have pseudopodia. These are having pseudopodia, due to which these are named as the podocyte cells. Podocyte cells are having pseudopodia with them. What actually they do is that they cause to help in the process of ultrafiltration, such that when the blood in the glomerulus comes, it gets ultrafiltrated. It gets all of the smaller particles in the blood get the filtered out of the capillaries, which are known as filtered out of the glomerulus into the Bowman's capsule, and from the Bowman's capsule these are then absorbed and then transported into the renal tubules. So for now, what actually happens is that the podocytes are having smaller slit-like openings. They are having smaller slit-like openings. Podocytes are having smaller slit-like openings, which are named as, which are basically 70 nanometer in diameter. 70 nanometer in diameter, as well as these internal slits are positively charged, due to which what actually causes is that um, um, whenever the blood is being filtered out into the Bowman's capsule from the glomerulus, then at that moment the at that moment, larger particles such as blood cells and plasma proteins are not supposed to come in the Bowman's capsule, due to which, in order to prevent them from entering into the Bowman's capsule, the podocytes are covered. The podocytes are having slit-like openings, which are 70 nanometer diameter and positively charged, which cause to repel the positively charged plasma protein, because plasma proteins are made up of basic amino acids, and as they are made of basic amino acids, they tend to get repelled by the positive charge of the holes, the slits or pores which are formed by the podocyte. As a result of which, the plasma proteins are repelled back from entering into the glomerulus, from entering into the Bowman's capsules from the glomerulus, and alongside that, it is prevented from entering into the Bowman's capsules from the glomerulus, alongside which the larger particles such as blood cells are not allowed to go, as the diameter is around 70 nanometer of the podocyte internal opening. Hence, the the podocyte basically prevent the passage of plasma proteins into the Bowman's capsule. And one more thing that I wanted to highlight over here is that it is also surrounded. The glomerulus is surrounded by podocytes, and the Bowman's capsule is surrounded by macula densa cells. Macula densa cells absorb, which cause to absorb the filtrate. The material which has been filtered out is known as filtrate, which is filtrate causes to absorb the filtrate in and send it into the proximal convoluted tubule or proximal convoluted tubule, or in broader terminology, the renal tubules. So this is the entire structure which is known as the Malpighian body or the renal corpuscle, right? And what actually drives the material to be ultrafiltrated into the capillaries is that the hydrostatic pressure which is created in the glomerulus. The hydrostatic pressure which is created in the glomerulus, also known as hydrostatic pressure which is created in the glomerulus, causes to um, drive the process of ultrafiltration or the glomerular filtration. In what actually happens is that, due to the difference in the diameter of afferent and efferent arterioles, afferent arterioles are having a larger diameter, efferent arterioles are having smaller diameter. Afferent arterioles are having smaller diameter, afferent arterioles are having smaller diameter, and efferent arterioles are having larger diameter, due to which it causes to create a pressure gradient. A gradient of pressure is created, which causes to generate hydrostatic pressure, and that hydrostatic pressure in the capillaries is directly proportional to the glomerular filtration rate, which causes the process of filtration to occur, right? And on the other hand side, if we talk about the afferent artery, is the one which is which is dividing into glomerulus, and glomerulus combine together once again to form an artery which exits an arteriole, sorry, which exists from the renal corpuscle, which is named as the efferent arteriole. And that efferent arteriole further is surrounded by the renal tubules, which we are going to discuss further. Renal tubules are basically consisted of four major regions. Number one is the proximal convoluted tubule. Proximal convoluted tubule is basically the major region where the absorption are basically renal tubules are the highly coiled region. These are highly coiled region where the process of reabsorption and tubular secretion occurs. Proximal convoluted tubule is a region. It is a region. Proximal convoluted tubule is a region where the reabsorption of most of the important and useful nutrients and most of the important material from the filtrate occurs. For example, glucose, amino acids, water, electrolytes are all basically the the materials which are filtered out of the proximal convoluted tubule are filtered are all which are filtered out of the proximal convoluted all of the materials which are filtered out of the proximal convoluted tubule. All of the materials which are filtered out of the proximal convoluted tubule. All of the materials which are filtered out of the proximal convoluted tubule. Filtered out is not a good word. We can say that reabsorbed selectively into the capillaries which surround the PCT. Now, these capillaries which surround the PCT are known as peritubular capillaries. The capillaries which surround the PCT are known as peritubular capillaries. We are going to discuss that as well. So there's a highly coiled region which is on PCT, and PCT after moving from PCT, the PCT goes down. The PCT goes down. The PCT basically goes down in the medullary region and extends to form a long loop. That loop is known as loop of Henle. That loop of Henle is basically divided into two parts: the descending loop of Henle, which is going downwards, and the ascending loop of Henle, which goes upwards. The descending loop of Henle is a bit thinner, and the ascending loop of Henle is a bit thicker. And on the other hand side, the pressure is higher in the ascending loop of Henle and lower in the descending loop of Henle. And after the loop of Henle, we've got the distal convoluted tubule, which is again found in the cortical region. And as you can see the diagram as well, this is cortex medulla junction. Upper region is in cortex and downward region is in medulla. So from the cortex, in the cortex we've got when the loop of Henle again comes back from the ascending loop of Henle, it is then transferred into the it is it then forms the highly coiled distal convoluted tubule, which is responsible for the mechanism which is responsible for the further reabsorption of important materials from the which is important for the further reabsorption of important materials from the distal convoluted tubules into the blood, as well as the reabsorption of urea also takes place. Reabsorption of urea also takes place in the distal convoluted tubule, because there is an entire mechanism behind it which we are going to discuss in the next video. Why urea is being actively reabsorbed, why is it being actively while we is it being actually selectively reabsorbed in distal convoluted tubule. There's a huge the mechanism behind it, but for now it's crucial to understand that it is in distal convoluted tubule, after which distal convoluted tubule ends up in the collecting duct. That distal convoluted tubule ends up in the collecting duct. The distal convoluted tubule ends up in the collecting duct, and that collecting duct again goes into the medullary region. From the medullary region it goes into the papillae, and from the papillae it goes into the minor calyx, and from minor calyx it goes into the major calyx, and from the major calyx it goes into the renal pelvis, and from the renal pelvis it goes into the ureter, and further through the urinary system it is then excreted out. And when it comes to the arterial system in nephron is in such a manner that first there is renal artery, which uh, divides into afferent arteriole, which is thicker in diameter, and the efferent arteriole, which is uh, thinner in diameter. Afferent arteriole is thicker in diameter, due to which it has got low pressure, and efferent arteriole is thinner in diameter, due to which it has got high pressure. As a result of the pressure gradient, there is a hydrostatic pressure created in glomerulus, due to which the ultrafiltration is possible to occur. After which the arteriole which exits the glomerulus is basically what we call as the arteriole which exits the glomerulus is basically what we call as the efferent arteriole, which further goes in the renal tubule region, and over there it divides into peritubular capillaries, which peri means near, tubular means near to the tubules, due to which peritubular capillaries are those capillaries which are near to the tubules. What their function is basically to provide nutrition to the oxygen. They provide nutrition as well as oxygen to the tubular region, the renal tubules, as well as they help in the process of selective reabsorption. These are the two major functions. And when they go into the loop of Henle, the peritubular capillaries further branch themselves into smaller capillaries, smaller vessels, which are known as vasa recta. Vasa recta are important for three major reasons. Number one is countercurrent mechanism. They play a role in countercurrent mechanism, which we are going to discuss. And after which we've got number two, that they provide nutrition and oxygen to the cells of loop of Henle. And on the third, they are important in producing, they are important in selectively reabsorbing the water it inside the loop of Henle. For which we've got another and hormonal secretions as well. For example, ADH, antidiuretic hormone, vasopressin from the posterior pituitary gland is released, which causes to help in this process. All right. And then when it comes to DCT, again from the vasa recta, the blood vessels form the peritubular capillaries around the DCT, where they provide nutrition and oxygen, and help in the process of selective reabsorption and tubular secretion. So in distal convoluted tubule, these are again important for tubular secretion, selective reabsorption, and the process of providing nutrition and oxygen to the cells of distal convoluted tubule. One more thing that I wanted to highlight over here, that vasa recta is parallel. Vasa recta is found in the medullary region, due to which it is on the posterior side, on the parallel, the on the posterior side. These are vertical. The vasa recta on the posterior side are basically, vasa recta is present on the posteriorly. It is connected to the veins of the medullary region. Posteriorly, or on the back side, they are connected to the veins of the medullary region. On the posterior side, on the back side of the medullary region, on the backside posterior side of the medullary region, these are connect on the back side or the posterior side of the medullary region. On the back side or the posterior side of the medullary region, these are connected to the venues. These are in contact with the venues, where they provide nutrition and oxygen to the medullary region, whereas the peritubular capillaries provide nutrition and oxygen to the cortex as well. In cortex, particularly PCT and TCT, right? So these are major things that I wanted to highlight. Um, and collecting duct is basically the duct where no process of selective reabsorption or tubular secretion occurs. It's the only final stage where tubular secretion occurs. Rest, the material is simply transported out of it into the papillae, and then minor calyx, then major calyx, then renal pelvis. So this is all about this video. Hopefully we will meet in another video.